Hi, we are back here, and saying we'll start with the second block of today's conference, and I will start presenting our first consortium partner, Pierre Pétriacq from University of Bordeaux. He is the head of the Bordeaux Metabolome, and he is a PhD in biology and then postdoc in biochemistry and manager of the biological mass spectrometry facility, GLOMICAVE. Since 2017, he has been associate professor in biochemistry at the University of Bordeaux, where he researches on the plant metabolomics and redox biology. He was appointed as director of the Bordeaux Metabolome Facility in December 2018 and a visiting professor at the University of Stuttgart in April 2019. He is also a fellow of the Higher Education in Academy of Sheffield and vice president of the French-speaking network of metabolomics and fluxomics. So Pierre, when you want.
Thank you, Biotza. Good morning, everyone. I'm very pleased to be presenting this last result with you today. So I titled my presentation, Disentangling Genotype-Phenotype Relationships Using Machine Learning. And so I'm from the University of Bordeaux, I work at INRAE, and there are some contributors from my INRAE colleagues, Georges, Sylvain, Sophie, Milena, and Coralie. So this presentation is in three parts. First, I'm going to brush up the concept of genotype-phenotype relationships and also machine learning. And then I'll finish by presenting three relevant snapshots of different sectors and use cases that we used in the project. So what is genotype-phenotype relationship? So in 2008, Benfey and Mitchell defined this concept, and they stated that mapping gene to the function is called the genotype to phenotype problem, where phenotype is whatever is changed in the organism when a gene's function is altered. So in fact, the relationship between genotype, that you can understand in the age of molecular biology as nucleic acid or peptide sequences, and phenotype, in other words, any trait from the molecular to organismal scale. And this relationship is essential across biological discipline. Advances in omics, such as next generation sequencing, proteomics, metabolomics, and so on, can enable large-scale mapping of genotype to phenotype. And this use parallel methods to measure specific traits across various scales of living system. And in fact, to reveal unprecedented relationships, predictive biology can now be key. In fact, this strategy uses input data like omics or other large scale or even targeted data, and output data such as responses like phenotype traits that could be growth, yield, resilience to stress, exposure to contaminants or global changes, and using this different type of data through artificial intelligence, we can provide predictive models, and these models can be subsequently used as recommendations or also diagnostic tools. It's particularly relevant in the case of complex organisms and systems like environment, being bacterial, animal, plant-based, human, or environmental. And it's important because systems biology can benefit omics data to decipher complex phenotype. And as a reminder, as the last omic brick, metabolomics is particularly insightful and informative, because it's closer to the phenotype and it can integrate influences from the environment as well. So how can we learn from the machine? So, there are a lot of different AI methods that are now being used. For example, artificial intelligence is a set of theories and techniques that aim to create computers that can mimic intelligence as humans. Example, visual perception, automatic reasoning, intelligent robots. Within AI, we have machine learning, which is a subset of techniques that can enable computers to learn from the data and improve their performance over time. As a subset of machine learning, there are deep learning techniques like artificial neural networks, which are algorithms designed to work like the human brain, in other words, and this could learn from large quantities of data. And of course, data science, disciplines that relate to collection, management, and data analysis, and within data science, omics that have been presented before. So the overall objective is to benefit from comprehensive multi-omics datasets and predict relationships between genotypes and phenotypes that relate to diverse fields of life sciences. So we perform predictive modeling, predictive biology based on modeling, of quantitative and qualitative phenotype traits using machine learning. So in other words, first we get the data, and you'll see throughout the conference that we can get data in different ways. Then we prepare the data. So, for example, we define what target value or class we want to predict, what is actually relevant and useful, can we represent it using knowledge graphs. Then we train models by using machine learning algorithms like linear models, partial square, supervector machine, deep learning, and so on. We test the results to verify, to validate the prediction and the classifications. And then once it's refined, that's to say, if we need to use only parts of the data or get more information on the identified markers, we can apply these models to business cases. So in terms of outcomes, we get predictive models for measurable phenotype using multi-omics data in different sectors: agrobiotech, environmental, and animal sectors. So the very first trait is about fruit quality. So for this case, we aimed at predicting fruit quality using predictive metabolomics to examine key physiological changes that can contribute to growth, ripening, and physiology by using 10 different fruit species. So we measured in parallel different traits. And the motivation is to get a better understanding of the developmental basis of fruit growth, physiology, and quality that can allow the identification of molecular targets for their improvement in the near future, which is important in the context of global change, for example, or the agroecological transition. So we used metabolite profile because metabolites are synthetic descriptors of cell and tissue physiology, because we know that their abundance is determined by complex regulatory networks that can involve transcriptional changes and enzymatic scales, as well as level of other metabolites. So if we take a look at our data in a very descriptive way by using multivariate statistics, we can see that metabolomics can reveal distinct patterns in terms of developmental of the 10 species. And another very important outcome is a challenge, actually, because when you compare very different species, you tend to get a lot of missing values in omics dataset. So we had to impute missing value by using other algorithm to make sense of this dataset. And regardless of this challenge, we were able to describe very nicely distinct patterns in the different fruit species with a clear impact of the developmental stage on the fruit metabolome. Now, using this very complex and rich data, we want to compare predictive models by trying to get as many models that we can get, and this allowed us to select the best metabolic predictors of fruit traits. So, for example, here I'm presenting just a generalized linear model, and you see it's already a very rich information with different imputation methods and accuracies in terms of R-square, so coefficient of determination, or accuracy for classes, and for different traits, the acidity, because people like to get fruit that are acid but not too much, so it's an important physiological aspect. Climacteric fruit, relative growth rate, is it a slow or fast growing fruit? As a stage, of course, the level of sugars, so the sweetness of the fruit, and the percentage of development. So you see, we can get very different capacities in predicting those traits. But what is very important is if we take the top 80% of these predictors represented here, we can reveal important predictive markers of the traits, which can be very specific to a trait, like, for example, slow versus fast, or the acidity, or the sugars, or we can also have less common, but still with a decent level of overlap, some markers that could be interesting for different traits. And that's a gold mine for breeders, because if you target this metabolism, you can get the next variety of fruits that could be produced and eaten by people, by the societies. Next use case is about bioenergy production, and this was in collaboration with our colleagues from University of Minho in Portugal. And the idea was to predict the ability of the microbiome of an anaerobic sludge digester to remove organics and produce methane, as Nuria explained just before. So AD for anaerobic sludge digestion relies on a hosting of myriad of microbes. In fact, the sludge that can form a complex microbial ecosystems whose microbes can play a distinct role. And if we understand this microbial community, it could be very great impact on the optimization of eco-friendly anaerobic digestion. Okay. In fact, some microbes can break down complex material into simple sugars and acids, whilst other microbes can cover this into building blocks for methane. And methanogens microbes can assemble these components into methane gas, which is a renewable energy source. So we benefited from samples from wastewater treatment plants in Portugal, from primary sludge within a primary clarifiers, and secondary sludge after biological treatment. In parallel, we measured biogas production, and we tried to model, based on data obtained from the samples, the production of methane. And eventually, we want to decrease the operational cost by targeting or revealing the best microbes to digest. So studying microorganisms in these systems can pose challenges because they are in constant interaction and they are limited in growth in lab settings. So we adopted meta-omics techniques for deeper insight. For instance, we use meta-taxonomics that can help identify microbe species and their abundance. And by using machine learning on these data, we explore the connection between the sludge microbial composition and the biomethod production. So on the right figure here, we have the coefficient of determination, R square, for several different algorithms in terms of machine learning. And we can see that we can get very nice prediction around 0.8 in terms of R square. So 80% of chance to predict the right microorganism. So now going even further, when we select these microbial players in biomethane production, it's super important because these models can prove highly beneficial as they precisely identify which organisms can exert the most significant influence on predicting the gas production. Example here in this table, you have the importance score of some of the different bacterium. So we can, let's say, engineer further the digester to improve their biomethane production. And finally, the last use case is about combining multiple omics data to unveil a bigger picture, okay? Because if we integrate multi-omics, we found that we can enhance phenotype prediction. This was made in collaboration with Allianz in France and Cerida in Spain. And we combined omics dataset to boost our ability to understand phenotype, such as precisely cattle fertility. So this case study examines embryo transfer, which aim at disseminating superior traits within a herd. For example, embryo transfer can involve transferring embryo from top-performing animals, such as those with high milk production or disease resistance or other traits that are interested for the producer, into the wombs of multiple other females. And this strategy allows even average cows to produce high-value calves. That's important. However, and there's a caveat, success rate in embryo transfer can fluctuate a lot due to different factors, such as embryo quality, recipient health, transfer procedures. So the key to getting more cows pregnant after embryo transfer is finding the right cows to receive the embryos. So right now, the existing methods, the current method can overlook healthy cows. So we must refine recipient selection methods. That's super important. And in fact, research revealed that cows with successful pregnancy can exhibit a distinct blood signature. So by identifying this signature, we can aim to enhance recipient selection and therefore increase calf birth. So that was the objective of this use case. And in that case, we performed predictive modeling on plasma blood samples from the cows, and we combine metabolomics and proteomics. We apply machine learning as well on this data to predict pregnancy diagnosis and calf birth. So on the right, this figure summarizes the predictive power of the developed model, and remarkably, combining metabolomics and proteomics, we can enhance the prediction. You see here a shift in the R-square or the accuracy. So in other words, this underscores that it's very beneficial to integrate diverse omics types for phenotype prediction. So as a take-home message, machine learning-based prediction can unveil genotype-phenotype relationships, and this is even more valid for complex traits, regardless of the sectors being considered. And combining omics data can improve predictive capacity to allow better interpretations. So thanks for your attention, and I'm very happy to take a question or two if we have time.
Yeah. If it's fine, we will take them after Raman's presentation, and we will take all the questions there in a block. Okay. So let's move to our next consortium partner, Raman Siarheyeu. He is a research scientist at NEC Laboratories Europe. Raman, when you want.
Hello, good morning. Thank you, Biotza, for introducing me. Today I would like to present the section related to multi-omics integration. However, we'll mostly speak about integrating text knowledge into the whole picture of the data sets. Why this is important? As from the previous speeches, normally we try to analyze different modality of omics data on the molecular level. However, there is also another source of valuable information that can provide some insights inside the whole data set, and we consider it together. And this source of information is called text data. However, to integrate the textual data is not so easy. First of all, because the computers do not understand the text exactly like we humans do, and also do mistakes. This is also relevant for the large language models. For instance, they have own limitations related to hallucinations and fact-checking. So here we decided to follow, to find the solution to this problem, and our priority was the specificity of the extracted concepts from the text, and there is a tool that can help us to do that. And this tool is called knowledge graphs. However, first, in order to get to the knowledge graph, we should understand what does it mean to analyze text and to mine information from text. Imagine that you have a sentence, exogenous T6P drastically upregulates TPS1 expression in nicotinada tabacus under low nitrogen. And that means that, first of all, you need to split this text into the different, into the separate words. If we speak about the sentence, and each word should be tagged what is the part of the speech and which words are really the concepts that we wish to know or that we wish to have in our integrated knowledge base. After that, we can, when we understood the concepts that are present in the text, in case we also manage to understand the relations between the concepts, we can build a knowledge graph that will bring together all the concepts and also bring together the relations from the text as well as from the well-structured and well-curated databases. So in the end, we can have a single source of information that comprises molecular entities, information from molecular databases, as well as the publications that can be used to check or to verify the research hypothesis. Now I would like to go to more details about how exactly we do that. So here within the project, we developed the data processing and text mining pipeline that starts with the scientific literature search and extraction from the internet. Then we select the relevant papers and we do the text mining with the relevant papers. In the end, we create the knowledge graph that comprises all the relevant concepts that we managed to detect in the publication abstract texts. Now, more detailed about how we do exactly the recognition of the entities in the text that we wish that we would like to have. Imagine that we have a sentence. Embryos showed high level of palmitic and stearic acid content. Probably the first thing is just to do a simple string search of what we wish to find. Let's say we wish to find whether we have publications that mention palmitic acid. However, in this case, such kind of approach will not be successful because the string search will not be able to identify the entities that are not together or that are such parts of a more complex multi-word pattern. That is why we used more advanced synthetic relationship analysis between the parts of the sentence. And using this type of dependency parsing, we can create the combinations of the words that comprise the whole concept, the biological concept. For instance, in this our example, we can create the different combinations like stearic acid context, stearic contents, or palmitic acid context, palmitic content, based on syntactic dependencies in the sentence, and also define the criteria like if there is a concept that matches our dictionary and this concept is the longest multi-word entity, we can report that we have a match. And this is the case in this example. Also, when we are analyzing the text, it's very common that using just a string search, we might miss the concept, but we may catch a false concept. For instance, in this sentence, some GTPases are inactive through binding guanosine nucleotide dissociation inhibitor. A string search will probably catch the concept of guanosine because it definitely would be in dictionary of the biologically relevant concepts. However, the sentence is about a different concept. It's about the guanoside nucleotide dissociation inhibitor. And in contrast, our approach also allows to catch such differences and to get the correct multi-vote entities. The second part is how to extract the relations between the concept. And here is a method that are called open information extraction. I will just illustrate what it means, but not go into the details. Imagine that you have a sentence in pancreas's FOXO1 regulates B cell formation. And in order to translate into the machine readable language, you need somehow to make a structure of it and to formalize. And in this science, you do it using so-called triplets. Each triplet consists of the subject, object, and the relation. In this case, FOXO1 is a subject, pancreas object, and what it does, normally it contains a verb, this is a relation. We can also extract different attributions to those triplets and relations that may specify the context. For example, in this sentence, the finding that was extracted from some study according to the results from our experiments. And for the project, we developed the pipeline that does this text analysis and text mining in order to identify such kind of open relations. And also we adopted it specifically to extracting the multi-omics concepts. So, and in the last part, I would like to demonstrate how it looks in the GLOMICAVE knowledge graph. Imagine that you wish to search the information related to pyrophosphates. And here in the knowledge graph, since we now know how to catch the entities from text, how to catch the relations, and how to bring everything all together with the data from the well-curated and structured source databases, now we can really answer different questions. For instance, what are the papers and what are the sentences where we can meet this concept? And you can get it from the knowledge graph. Another question can be, okay, what are the synonyms for this concept? And what are the molecular entities that are tagged in different databases related to this concept? We can also get it from the knowledge graph. Or the questions like, what are the pathways of biological processes with this molecule also part of it? And we can get it from the knowledge graph. Same we can do with extracted open relations and facts, and also traverse up to the sentence where we met this concept. So the message that we wish to deliver here, that the knowledge graph is a really very powerful tool for converting structured text, unstructured text into structured knowledge that then can be easily understood by both humans and machines. And this allows us to integrate really a lot of text mine concepts and link them to well-established and curated structural data. So here we also developed our own approach how to extract the fine-grained concepts from the text and incorporate them into the knowledge graph. And this knowledge graph currently is the basis for this search on the GLOMICAVE portal, and we are trying to understand whether we have some support of the hypothesis made by research in the literature. So this is all from my side. Thank you very much for your attention.
Time to couple of questions that we have received from the audience. It refers to no, you mentioned that no information was extracted from abstracts from papers, and the question was, what is the impact of restricting your research to the abstracts, no, compared to the full text?
Okay, it's a good question. There are two points that I should mention. First point is legal. So legally, you are not allowed to use the content of the whole paper in most of cases, because this is a property of the publisher. The second is the scalability of the method. So to work with the whole text is much more complicated than to work with the abstract. However, the abstract normally, if it's a good written abstract, already contains the most important conclusion from the paper.
Thank you, Roman. So now, Nuri, a couple of questions, more, I would say, more general regarding the GLOMICAVE. There are a couple of them, but just I will mention some of them, and then I'll say, no, we will send the others reply. But for instance, if this if GLOMICAVE can be used, or it's applicable to other fields or other cases. I know, Nuria, and yes, also a little bit if support human genotype, phenotype analysis.
Yeah, well, GLOMICAVE is designed not for human genotype, phenotype, or disease relationships, because the scope of the call excluded. So we focused on different fields, so in animals, or vegetables, and environment. But yes, and we want to improve on these fields our tools. So next month, next years, introducing more information in the knowledge base related to other species, animals or vegetal species, and also to improve tools to work better with microbials. And but in the field of human, it's a very interesting field. So we have seen that there are a lot of tools related to that, and for sure we will be interested on to develop another similar platform, but maybe will be not GLOMICAVE, will be another one. Yeah.
So thank you again, Pierre, Roman, and Nuria for the presentations, for getting into GLOMICAVE. And we just maybe break 5 minutes for a quick coffee, and then we start again with the next session. Thank you very much.